All right, welcome to the second lecture from chapter 16, which is all about glycogen metabolism and gluconeogenesis. In this lecture, we are going to be discussing all about glycogen metabolism. So in this chapter, in the first part, we talked about gluconeogenesis, and now in the second part, we're going to discuss glycogen metabolism. First, we'll talk about glycogen breakdown, and then we'll discuss glycogen synthesis, the opposite of breakdown, and then finally we'll talk about how both of these processes are regulated. So again, we have our overall glucose metabolism pathway diagram shown here. And here we're going to discuss, like I just mentioned, glycogen synthesis and breakdown. So here we have glucose being built up into glycogen, that's glycogen synthesis. And when glycogen is broken down to yield glucose, that's glycogen breakdown. And glycogen, as we should know already, is the essentially storage form of glucose, that highly branched chain version of glucose. So let's now review the glycogen structure. So we know that glycogen starts out with some linear chains of glucose. All the linear chains are connected by alpha-1,4 glycosidic bonds, which are illustrated here. So in this glucose on the left, we have the anomeric carbon. Carbon 1 is in the alpha configuration, and it's connected to the 4 carbon of the glucose that is on its right, hence the alpha-1,4 glycosidic bond. There are also branch points every 8 to 14 glucose monomers, and the branch points are connected by alpha-1,6 glycosidic bonds. We have that illustrated on the bottom here, where we have the 6 carbon of the glucose on the bottom that is connected to the 1 carbon of the glucose shown above it. And that's the anomeric carbon of that glucose, which is again in the alpha configuration. And it's because it's attached to the 6 carbon via the branch, that's an alpha-1,6 branch point. So breakdown of glycogen can occur simultaneously from all of the non-reducing ends. And essentially, almost all the ends of glycogen are non-reducing ends. They have an anomeric carbon, which has the ability to be reduced. There's only one reducing end in glycogen, and that's the one that's in the center of the molecule that doesn't have an end available. So let's talk a little bit more about glycogen breakdown. So another, maybe more technical term for glycogen breakdown is glycogenolysis. But we can just refer to it as glycogen breakdown. Most of the glycogen in our bodies is found in the liver as well as the muscles. In the muscle, glucose 6-phosphate is used locally for glycolysis for a quick source of energy, particularly during bursts of exercise. On the other hand, in the liver, when glycogen is broken down into glucose 6-phosphate, it is then converted to glucose and exported for use throughout the body as a fuel. So for glycogen breakdown, for this to happen, 3 enzymes are required. There's glycogen phosphorylase, which we learned a lot about during chapter 12. And then there's also debranching enzyme, as well as phosphoglucomutase. And all of them are shown in this diagram on the right here. So we have phosphoglucomutase, glycogen phosphorylase, and the debranching enzyme. And so phosphoglucomutase is involved in the conversion of glucose 1-phosphate to glucose 6-phosphate because glucose 6-phosphate can be used for glycolysis, but glucose 1-phosphate is the breakdown product of glycogen phosphorylase. And debranching enzyme is really important for taking apart those branches because glycogen phosphorylase is only breaking those alpha-1,4 glycosidic bonds. Debranching enzyme is necessary to help with those alpha-1,6 branch points. So first we'll do a little review of glycogen phosphorylase, which we already learned about a little bit in chapter 12, particularly the regulation of it. So the overall reaction that's catalyzed by glycogen phosphorylase is glycogen containing N residues plus 1 inorganic phosphate yields glycogen with missing glycogen with missing 1 glucose residue plus 1 glucose 6-phosphate. And it's a phosphorylase because it is this enzyme is breaking a bond using phosphate as the attacking group, which is ultimately getting attached covalently to that product, the 1 glucose monomer. And like we mentioned a slide or two ago, glycogen phosphorylase is going to remove glucose residues from all of the non-reducing ends of glycogen. And this can happen from all of the ends at once. And this is part of why, as we discussed previously, there are so many branch points in glycogen, because the more branches you have, the more ends you have that can be broken down very quickly when glucose is needed. And like we also mentioned, glycogen phosphorylase is going to break those alpha-1,4 glycosidic bonds that make those linear chains of glucose. However, glycogen phosphorylase actually stops about 4 monomers away from any branch point because the branch doesn't physically fit inside the enzyme. And this is called the limit branch. And we also had discussed a lot in chapter 12 about how glycogen phosphorylase itself is a highly regulated enzyme. And we'll talk more about that process a little bit towards the end of this lecture as well. So next, let's discuss glycogen debranching enzyme. So glycogen debranching enzyme gets involved once you hit that limit branch where glycogen phosphorylase kind of gets stuck. So what the debranching enzyme does is first it moves the last 3 glucoses, which are shown here in red. And these are all connected by those alpha-1,4 glycosidic bonds. They are moved over to one of the other linear branches of glycogen. And after the debranching enzyme does this, this ultimately makes one of the branches longer so that glycogen phosphorylase can again start to break it down 1 glucose at a time. The debranching enzyme, which you see over here, we have this longer branch that glycogen phosphorylase can now take care of. On the other hand, now we still have this 1 glucose left, shown in green, that's connected by that alpha-1,6 glycosidic bond. And so glycogen debranching enzyme also is going to hydrolyze this 1 last glucose into a glucose monomer, and it's just glucose. It doesn't make it into glucose 1-phosphate. So ultimately, and this is a hydrolysis process, not a phosphorylasis process. So ultimately, when glycogen is broken down, about 90% of that breakdown yields glucose 1-phosphate monomers, and about 10% just plain old glucose monomers because of how highly branched it is. So every single branch point has that 1 last glucose that is going to get broken off as a glucose. And finally, let's talk about phosphoglucomutase. So once all of those glucoses are broken off using glycogen phosphorylase as glucose 1-phosphate, phosphoglucomutase helps to convert glucose 1-phosphate into glucose 6-phosphate. And once you have glucose 6-phosphate, which is shown also down here, now this can be used as an entry straight into glycolysis in step 2. Or it can be converted back all the way into glucose to be exported. Because it is phosphorylated, well, both these molecules are phosphorylated, they're charged, which makes them very difficult to pass in and out of cell membranes. And so by dephosphorylating them into plain old glucose, they're able to more easily pass in and out of cells through glucose transporters. Oops. And so, yeah, okay. So that brings us to the other end, right? We already discussed the 3 enzymes involved in glycogen breakdown into glucose 1-phosphate or glucose 6-phosphate. So now let's talk about the reverse, which is glycogen synthesis. So that's going to also involve a few different enzymes. Phosphoglucomutase, the enzyme that helps to interconvert glucose 1-phosphate to glucose 6-phosphate, this reaction is reversible. It's pretty clear equilibrium, so this is a reversible reaction. So phosphoglucomutase is used to catalyze this reaction in both directions. However, the other 2 enzymes that are used to break down glycogen catalyze irreversible reactions. Those were debranching enzyme and glycogen phosphorylase. So we're going to have to, or the cell is going to have to use different enzymes to help build up glucose 1-phosphate back into glycogen. And so it is not the exact opposite of glycogen breakdown. And it's also going to require some energy input. So again, this is something that you're only going to want to happen when energy levels in the cell are high, or sugar levels are high, because you want to store up that energy, and it's going to also use energy to make it happen. So this, we'll talk more about the regulation later, but sort of keep that in mind as we go through this. And so there are a few different enzymes involved in glycogen synthesis. And let's go through them one at a time. So first we have UDP glucose pyrophosphorylase. This is a mouthful, but we can still help to use the name of it to help figure out what it does. So we have a pyrophosphorylase. This name hopefully sounds a little bit familiar. If you think of just a phosphorylase, we know what that does, right? That's going to use phosphate as an attacking group to help break a bond and adding on a phosphate group covalently in the process. A pyrophosphorylase would have the same function, except instead of using one inorganic phosphate, it uses inorganic pyrophosphate. And if you look at the reverse of this reaction, that is what you would see. You have UDP glucose here with this high energy phosphate bond. You can imagine adding a pyrophosphate there as an attacking group, which would break off, which would help to make UTP as one product and glucose 1-phosphate as the other product. However, this is another one of those enzymes that's named for the reverse reaction, because we're actually seeing this in the other direction, right? So what's happening here with UDP glucose pyrophosphorylase is it has substrates glucose 1-phosphate and UTP. And the products of it are UDP glucose, or UDPG, and a pyrophosphate, an inorganic pyrophosphate. And this ultimately is sort of helping to activate the glucose by putting it, by adding it onto this really high energy phosphate bond. And the pyrophosphate itself also has this really nice high energy phosphate bond in it that we're going to see is actually going to help produce energy to help drive the rest of glycogen synthesis. And that's what we see in the next step. In the next step, inorganic pyrophosphate is hydrolyzed by an inorganic pyrophosphatase enzyme, and that's going to be made into 2 inorganic phosphates. And this is going to, that helps to drive that reaction that we discussed. That is the conversion of glucose 1-phosphate and UTP into that UDPG final product. And this helps to make that reaction spontaneous. And the overall delta G of this whole reaction is about negative 19 kilojoules per mole. So that is the sort of first part for glycogen synthesis, where we've the first sort of part of glycogen synthesis where we've activated our glucose. Now that activated glucose is ready to be added on to a growing glycogen molecule. And the enzyme that does that is glycogen synthase. So glycogen synthase is going to add alpha-1,4 glycosidic bonds. And so in some ways, this is sort of like the reverse of glycogen phosphorylase, because that's breaking alpha-1,4 glycosidic bonds. However, this is not, in this direction with glycogen synthase, we're not using phosphate as an attacking group. We already have our activated glucose, or UDP glucose. And so glycogen synthase is going to use the high energy phosphate bond that's already contained in the UDP glucose to help drive this synthesis of glycogen by adding 1 glucose residue to it and cutting off the rest of the UDP molecule. So the net effect of this is that we've converted 1 UTP into 1 UDP. So we've essentially consumed 1 phosphate, high energy phosphate bond. And that's for every glucose that eventually gets stored in that larger glycogen molecule. The other aspect of glycogen, as you know, is we have to make all those branch points, right? So glycogen synthase is only able to help make those alpha-1,4 glycosidic bonds. So glycogen branching enzyme is needed to help make those alpha-1,6 bonds to make those branches. So what glycogen branching enzyme does is it will take the last 7 glucose monomers from a linear chain, and it moves them onto one of the other glucoses in that large glycogen molecule and creates an alpha-1,6 bond there to create a branch. So here we have seen in red these 7 glucose monomers. They're broken off from that alpha-1,4 linear chain and used to attach somewhere else on that linear portion as an alpha-1,6 branch point. So that brings us, now we know that glycogen, how all the enzymes work, how it's built up, and how it's broken down. Now let's talk a little bit more how glycogen metabolism is controlled. So glycogen metabolism, just like we saw for glycolysis and gluconeogenesis, the regulation of it is going to happen at the irreversible steps, which are different in the 2 pathways. And for the same reasons as we saw for glycolysis and gluconeogenesis, because the reversible steps, if you speed them up or slow them down, that's going to affect both directions equally. It's the irreversible steps that help to control one pathway differently from the other pathway. So for the case of glycogen metabolism, the 2 irreversible reactions that are used for control are glycogen phosphorylase and glycogen synthase. And just kind of like we saw with control of glycolysis versus gluconeogenesis, the factors that are going to activate glycogen phosphorylase for breaking down glucose are also going to deactivate or inhibit glycogen synthase, and the opposite. And just like we saw for our other pathway for glucose, the control of these two enzymes can be allosteric, covalent, and hormonal. And we already talked a lot about glycogen phosphorylase, where it had these allosteric effectors. So AMP, which was indicative of low energy levels, helped to activate glycogen phosphorylase to help free up glucose to be used in glycolysis to eventually create more energy currency for the cell. And then we had those allosteric inhibitors, glucose 6-phosphate, glucose, and ATP, which all indicated either high glucose levels or high energy levels, indicating that glycogen does not need to be broken down because there's already sufficient energy or glucose available. On the other hand, in the opposite, we have the allosteric effectors of glycogen synthase. And the one main one here we have is glucose 6-phosphate, which activates glycogen synthase. So if we have a lot of glucose 6-phosphate, that's indicative of high glucose levels, which would be a good time for the cell to start building up glycogen as a storage form rather than using up glucose when there's a need, because there's not a need to break it down as energy when there's high glucose 6-phosphate levels. So let's look a little bit more about that control and the hormonal as well as the covalent control of these enzymes. So first we'll start with glycogen breakdown, which as we know is going to involve control of glycogen phosphorylase. So glucagon is the hormone that tends to be released when there are relatively low energy levels and low levels of blood sugar. This hormone, as we discussed previously, is going to ultimately activate protein kinase A, which is the kinase that's going to add phosphate groups to its substrates. And one of the substrates for protein kinase A is the enzyme phosphorylase kinase. When phosphorylase kinase is phosphorylated by pKa, it's active. And when phosphorylase kinase is active, it's going to phosphorylate its substrate, which is glycogen phosphorylase. And as we know, when glycogen phosphorylase gets phosphorylated by phosphorylase kinase, it is also active. And when glycogen phosphorylase is active, it is going to help break down glycogen. And this is going to happen, like we said, due to glucagon signaling when there are essentially low energy levels in the body or low blood sugar levels. On the other hand, insulin signaling occurs when there are high levels of blood sugar. Insulin is going to eventually activate a enzyme called phosphoprotein phosphatase 1, or PP1. And we know phosphatases are going to do the opposite of kinases. They remove phosphate groups. So PP1, phosphoprotein phosphatase 1, is going to dephosphorylate, and its substrates are both of these other enzymes, phosphorylase kinase and glycogen phosphorylase. Both of these are substrates of PP1. So after insulin signaling, these enzymes are going to get dephosphorylated. And ultimately, that's going to make both glycogen phosphorylase and phosphorylase kinase less active. And when glycogen phosphorylase is less active, this means there's no glycogen breakdown, which makes sense because when there is insulin signaling, there is high levels of sugar available. There's no need to break down glycogen because there's no need for more glucose. There's already plenty of glucose. So let's look at how that works in terms of glycogen synthesis. So once again, we'll start with glucagon, indicative of low energy levels. It's going to once again activate protein kinase A, which is going to phosphorylate phosphorylase kinase, which is going to phosphorylate glycogen synthase. Unlike glycogen phosphorylase, when glycogen synthase is phosphorylated, it's less active. And so this is going to mean there's no glycogen synthesis, which makes sense. When there's no low energy levels or low glucose levels, there's no substrate, so there's no need to make glycogen. On the other hand, during insulin signaling, when there's high blood sugar levels and high energy levels, phosphoprotein phosphatase 1 is activated. It's going to dephosphorylate both of these enzymes. And when they're dephosphorylated, that means glycogen synthase glycogen synthase is now going to be active. And when glycogen synthase is active, that's going to promote glycogen synthesis. So ultimately, when there are high blood sugar levels and there's insulin signaling, that is the time to start building up glycogen because there's lots of extra glucose around. It's a good time to store it so that it can be used later when there are lower glucose levels. And so hopefully these two things, you can see how they're sort of controlled oppositely. Glucagon will activate glycogen phosphorylase, but insulin will activate glycogen synthase because these types of pathways are needed during sort of the opposite conditions in terms of energy levels and sugar levels in the cell. And then to look at the bigger picture, we've sort of looked at this diagram before, and now hopefully you have a better idea of what's happening during glucagon signaling, which in the liver is helping to promote glycogen degradation, as well as gluconeogenesis. On the other hand, during insulin signaling, we have activation of glycogen synthesis, both in the liver as well as in the muscle. So hopefully this will sort of give you a sense now of how all these things are connected to each other. And as you go on to study all of this, it might be helpful to sort of think about what similarities are in between all these different pathways we've learned for glucose metabolism. And then you can sort of learn by thinking about what the differences are instead of sort of thinking about each one as a separate and distinct process, because they are really all very closely related to each other. So with that, now you should be ready to answer the Chapter 16 Glycogen Metabolism practice questions.